Okay, welcome everybody. We're going to get started here. Thank you for joining us for our Knauss Fellow Seminar Series. We have our first speaker, Andie Chan. She is working as a fellow for NOAA Fisheries Office of Science and Technology, working as a fisheries science coordinator for the National Stock Assessment Program, as well as the National Observer Program. Andie has been working on bycatch-related issues and incorporating socioeconomic information in fisheries stock assessments in that role. And prior to that, she recently graduated from Penn State with a Ph.D. in ecology, where she studied the effects of climate change on coral symbiosis.
Nailed it.
And she is now going to tell us a bit more about that work and talk to us about climate change and coral reproduction.
Thanks, Casey. Oh, dark. Thank you, everybody, for coming to my talk. I have to apologize in advance. I've been a little sick this week, so when I was putting this talk together, I hope it all makes sense. But if something doesn't make sense, please do let me know. So let's jump right in. Oh, maybe not. Is this working? Work now? Okay, perfect. Okay, so climate is changing. I don't need to tell this group of people this. It's causing all sorts of negative consequences for human populations, such as coastal flooding, more intense tropical storms, which is damaging a lot of our human habitats, and also ocean acidification, which is also decreasing fisheries productivity. But climate change is also bad for coral populations. So something that most people have heard a lot about is coral bleaching. So when corals bleach, the algae that's in their tissues leaves the coral cells, and the algae is important for providing food to the corals. And so if that algae remains outside of the corals, then they starve. And so we're losing coral individuals this way. There's also been increasing disease events that are causing a loss of coral individuals. So corals are dying from disease, including the stony coral tissue loss disease that some of you might have heard about that is sweeping through the Caribbean. And also more intense tropical storms are removing coral cover from reef environments. So we're losing coral individuals in all of these ways from populations. So if we want to understand how corals could recover, we need to understand coral reproduction. And this is important to understand because corals provide a lot of important ecosystem services, such as protection of coastlines from wave actions and storms, tourism revenue, and also habitat for commercially important fish species. So, coral reproduction. So corals reproduce in two ways. Corals can reproduce sexually, where different colonies will release eggs and sperm into the water column. Those eggs and sperm will rise to the surface and can combine to form coral larvae, which will swim a distance and settle and metamorphose to become a coral primary polyp and become a new coral individual. Corals can also reproduce asexually, where a piece of that coral can break off and move some distance and then reattach. So these are two ways that corals can actually input individuals into the population. And some terminology that I'm going to use throughout this talk is genet versus rammet. So coral genets are unique genotypes. So for example, all of us are different genets in this room. We're all unique genotypes, whereas coral rammets belong to the same genet. So when pieces break off and you have clones, those are called rammets. So climate change is expected to impact both of these processes. Considerably more focus has been paid to sexual reproduction because it creates new combinations of genes or new genotypes, and that's an important process that we talk about when maintaining populations. So some ways that climate change can affect coral sexual reproduction include reduced fertilization success, embryonic abnormalities, so we're getting fewer embryos surviving to larval stage, the faster development of coral larvae, which has strong implications for the connectivity of populations in the future, and also reduced settlement success. So this ranges from little effect on coral species to like strong negative effects in terms of climate change impacts on coral sexual reproduction. But less attention has been paid to coral asexual reproduction. So how is climate change going to impact this process of fragmentation and attachment of colonies, which still doesn't create new genotypes, but it can maintain genotypic diversity in the population and can increase coral cover. So it's still a very important process that we need to understand when we're talking about climate change effects. And some important impacts on coral asexual reproduction that are expected to change with climate change include increased storm intensity. So with more intense storms, we're expecting more fragments to be generated, so that could lead to more coral asexual reproduction. We're also expecting reduced coral skeletal density, and this is primarily from ocean acidification effects, where the skeleton will be more porous and more easily break. And so that could also contribute to more asexual reproduction, and also increased bioerosion. So when I talk about bioerosion, I'm talking about the invertebrate species that actually burrow into coral skeletons. And so they're removing large, I guess, trajectories, I don't know what word I'm thinking of, burrows of coral skeleton, and then corals will often fragment on that burrow. So that also makes corals more likely to break. And bioerosion is expected to increase with climate change, both because of ocean acidification, it could facilitate burrowing processes, and also eutrophication. So when there's more nutrients in the water, there's more primary productivity, there's more plankton for those bioeroding organisms to eat, their populations are going to boom. So I've been talking about genotypic diversity already, so I wanted to give you a quick primer on genotypic diversity. So this image here shows a happy population of people, all with unique genotypes. So there's high sexual reproduction relative to asexual reproduction. So there's high genotypic diversity. In this bottom image here, there's a lot of evil clones, but there's low sexual reproduction relative to asexual reproduction. So there's low genotypic diversity. And like I said earlier, this could be good for increasing coral cover in an area, so we could still get some of those ecosystem services that coral reefs provide, but it could be also bad if stressor comes through. So low genotypic diversity can also mean higher susceptibility to stressors. And a prime example of this was the Irish potato famine, where they're in Ireland in the 1840s, they're primarily planting one clone of the Irish lumper potato, which had the same susceptibility to blight. And so huge die-off of those potatoes, a lot of people starved. If they had been planting more diverse potatoes, then they could have had diverse susceptibilities to that blight. Some of them could have survived. We would have had higher resilience to that disease. This has also been shown in experiments with seagrass beds, where people have manipulated the genotypic diversity of seagrass beds and subjected them to some stressor, could be grazing, could be temperature stress, and measured recovery. And the more diverse seagrass beds were able to recover faster. So this genotypic diversity is clearly important, and we want to understand it more in coral reefs. So this returns me to my original question, is how will climate change impact asexual reproduction in reef-building corals, and how does that play in with the genotypic diversity of corals on the reef? Oh, so my focal species for this study was Parietes lobata, or the massive lobe coral. This is a very important reef-building species in many parts of the Pacific, and it's a broadcast spawning coral, so it releases its eggs and sperm into the water column. It's also very widely distributed and one of the most common coral species in the world. So we wanted to see how asexual reproduction in the species is responding to climate change. So in order to do that, we looked at two gradients in ocean acidification in Palau and the Galapagos, and we looked at how coral genotypic diversity varied along those gradients, and also looked at factors that could be influencing that genotypic diversity. So I'm going to talk a little bit more about those acidification gradients. So this is a busy slide, so give me 1 second. So on the left, there's a map of the Galapagos, and we have different islands here, and it's sort of split into the northern and southern islands. And the northern islands are less acidic, so that's the graph we have here, which shows the islands on the x-axis, aragonite saturation state. On the y-axis, which shows you how acidic that site is. So the aragonite saturation state is higher, it's less acidic. So the northern islands are less acidic than the southern islands. And in Palau, we have a similar gradient in acidification. However, it's not north to south, it's inshore to offshore. So it's kind of hard to see in this tiny figure here, but there's inshore sites here labeled 7, 8, and 9, and those on this corresponding aragonite saturation graph, 7, 8, and 9 are with lower aragonite saturation state, meaning they are more acidic. So we sampled along both of these gradients from north to south in the Galapagos and inshore to offshore in Palau. So really quickly, I'm going to go through some of our brief methods. Like I said earlier, we wanted to look at the factors that could be affecting asexual reproduction in this species. So we wanted to look at the frequency of large storms that could come through and break these corals apart. We wanted to look at the density of our coral skeletons to see, if they're maybe more porous in the more acidic areas. And we also measured mussel density per coral area using these photographs. So this photograph is showing you there's like little black holes throughout this coral, and those are all mussel boreholes. So we used photographs to trace the area of that coral and also count the mussel boreholes and calculated a mussel density. So that was our measure of bioerosion. And we genotyped our coral colonies using 10 microsatellite markers to create multilocus genotypes. So we were able to tell clones from unique individuals, so ramets from genets, and we calculated metrics of genotypic diversity and evenness. Okay, so a little plug for NOAA here. We looked at the NOAA historical hurricane tracks database to compare the amount of hurricanes going through the Galapagos versus Palau. And what we found was that there were essentially no hurricanes going through Galapagos and 208 hurricanes going through Palau since 1885. So if hurricanes were the primary fragmentation force driving asexual reproduction in the system, we would expect to see really low genotypic diversity in Palau compared to the Galapagos, since there's more of this fragment generating force there. So we also looked at coral skeletal density. So these measurements were provided by our collaborator, Derek Manzello. So if you have specific questions about how the coral skeletal densities were measured, please don't ask me. So this is a graph showing our skeletal density results. So our sites are on the x-axis, and on the left side, there's a dotted line here. The left side is the Galapagos, the right side is Palau, and these gradients on the bottom show the aragonite saturation state. So again, the high aragonite saturation state are the less acidic sites, low aragonite saturation state is more acidic, and the same here for Palau. And on the y-axis, we have our skeletal density. I'm sorry, online people, still trying to come over the sickness. So generally what we're seeing is that skeletal density decreased in the more acidic sites. There's definitely some variability there. So if skeletal density is primarily driving our differences in coral asexual reproduction, we would expect to see lower coral genotypic diversity at the more acidic sites in the Galapagos, so that's the southern Galapagos, and in Palau, so that's inshore Palau sites. And then, as I mentioned earlier, we also looked at the bioerosion. So we expect bioerosion to increase with climate change. So we expected our mussel densities to be higher in those more acidic sites in the Galapagos and Palau. Indeed, that is what we found. So our mussel density increased in those more acidic sites. So this is the same orientation as the previous graph with our site on the x-axis and our mussel density on the y-axis. And we generally see an increase in mussel density in the more acidic Galapagos sites and also in the more acidic inshore Palau sites. So we would expect to see a similar pattern in coral genotypic diversity from in the increased bioerosion and also the decreased skeletal density, where we'd have lower genotypic diversity in those more acidic sites. Oh, this is supposed to be like, well, what did we actually find for the corals slide? So this is a little busy. So these are polar plots, and so they show the spatial distribution of the coral colonies. So we sampled them in a circular way so that we can see their positions relative to one another. And each symbol is a coral that we sampled, and the color corresponds to the genet, or the unique genotype of the coral. And so if two symbols have the same color, like this one here, then those are ramets of the same genet, meaning they're clones. So they have the same genotype. So this is the northern Galapagos, which is the less acidic site, and this is the southern Galapagos, more acidic. So as you can see, there's lower genotypic diversity in the southern Galapagos. So this is great. This is what we expected from our bioerosion results and from our skeletal density results. I was really excited when I got this result. Wish I could have stayed here. Then we went to Palau, and we found, despite the similar abiotic conditions, similar bioerosion levels, skeletal density, in the more acidic Palau sites, we found the same high genotypic diversity in both inshore Palau and offshore Palau. And so we're trying to understand what could be driving this difference in pattern. So this is just another way of looking at this data. So this is genotypic diversity and genotypic evenness on the y-axis here. And evenness takes into account sort of the number of ramets per genet, sort of like how you think about evenness in species metrics. So again, the southern Galapagos falls out as very low genotypic diversity and evenness, whereas the Palau sites are all relatively high. So what is driving this difference in coral asexual reproduction in these sites? The short answer is, I don't really know, but I have a hypothesis for you, and maybe someone in the audience will have some great ideas. So Galapagos and Palau might represent ends of a spectrum. We would definitely want to look at asexual reproduction levels in more areas around the world, and not just these two sites. So these interactions with other species might be really important. So that this is just a graph of reef fish species richness. So the colored dots indicate the richness of species. So more red colors are higher number of species, and more green colors are lower numbers. So in the Galapagos, there's fewer fish species overall than in Palau, and we think maybe these interactions with fish species could be driving these differences. So what do I exactly mean by this? So a former student in the lab working in the Eastern Tropical Pacific found that these triggerfish species actually prey on the mussels inside these mounding corals. Well, they'll bite all these coral heads and eat the mussels out of it and generate all these rolling stone fragments. That's what they're called, which can move some distance and reattach, whereas like a single fish could create something like 70 fragments in just a short amount of time. So we're thinking that it's possible that these fish are operating in the southern Galapagos, where there's maybe not as much food sources for them, and so they're choosing to bite into these corals to get at these mussels. Of course, we don't actually have the data for this, so it'd be a really interesting interaction to explore. Whereas in Palau, the populations are maintained primarily by sexual reproduction, so we're having new larvae come. These are coral larvae, like obviously not to scale, that come in and can settle and make new colonies this way. So it seems like there's different processes driving reproduction in these areas, and really understanding the interactions with other species would be really informative. And so my conclusions for this are that multiple regions should be considered when studying processes like asexual reproduction and Paredes lobata. So if I'd only looked at, the Palau or Galapagos, I would have had a very different story. So what we really need to do is actually survey more areas if we want to understand how we can predict climate change will impact asexual reproduction in this coral species and in other coral species. And despite high bioerosion in the acidic Palau sites, genotypic diversity remain high. So there's no fragment generating source, or those fragments aren't surviving in those inshore Palau sites. Not exactly sure what is driving that exactly, but it's an interesting area for future research. Sexual reproduction seems to be absent from the southern Galapagos, so this population has reduced resilience. And this is really important for conserving these corals into the future. If a stressor wipes through that area, it's unlikely that those populations will survive, since there's very few genotypes with different resiliences. And bioerosion is expected to increase in the future, and this is possibly going to cause increased asexual reproduction in some areas of Paradis lobata's range, but not in others. So it's very context dependent, and we really need to understand those interactions. And again, understanding how fish that prey on those mussels interact with corals throughout the range would be informative for really teasing apart the drivers of sexual and asexual reproduction in the species. Oh, and then I didn't do all this work myself, so I would like to thank my co-authors, my former lab, people who provided field support, lots of undergraduates who counted a lot of mussels in photos, and my funding sources. And I'll take any questions. Thanks.
Thank you, Andie. And for people who have questions, just raise your hand and Casey will bring the mic to you.
Hey, interesting presentation. I'm curious, did you, in the Galapagos, in addition to north-south gradients, did you look at east-west gradients? Because the west side, where Arvina Bay is really different than San Cristobal because of upwelling.
You know, we didn't look at we didn't sample in an east-west fashion, but that's definitely true. There's a lot of different geochemical processes going on there, and I would probably talk to my collaborator Derek, since he's the Galapagos expert. But that's really interesting to think about for sure.
Anyone else in the room have questions?
My question's more related to if you've considered disturbance theory as far as helping to structure the communities there, given that the Galapagos seems to be fairly stable system, especially if you look at hurricane as a disturbance, but maybe related to other disturbances, and that might allow the clonal approach similar to the reverse of the potato famine to take hold versus a place that's more disturbed like Palau.
Yeah. So that could be really interesting in terms of like clearing space. So like in terms of survival of a larval coral that's settling, they're very small when they settle versus an asexually generated fragment. The asexually generated fragment would have higher survivability probability. So it's possible that those hurricanes clear space and that the coral larva can then settle. So that's an interesting way to interpret that, but I'm not sure if that's exactly what's going on. Does that kind of get at what you were saying?
Yeah, I could see that angle, but also maybe in the sense of what the species composition is in the area, that it would allow more of one species to become a dominant in the area in a stable environment versus something coming through and reshuffling the deck.
Yeah, like Paradis lobata is a climax species, kind of. Yeah, they are typically a very hardy species in terms of temperature tolerance and acidification tolerance. So that definitely, it is a marginal environment. So that's, in terms of species composition, could be what's going on. I'm not sure if that's what is driving genotypic diversity though, like within the species.
Any other questions here? Okay, so we don't have any online questions at this time, so we'll just leave it there for now and pick it up again with Ali at 12:30. So thank you, Andie.
Ooh.